In this video, topic 1.5, we'll discuss the structure and function of biological macromolecules. DNA is a macromolecule built from monomers. The monomer unit of DNA is called a nucleotide. Nucleotides have three components: the phosphate, the sugar, and the base. Linking these monomers together, that is a dehydration synthesis reaction or a condensation reaction, forms a sugar-phosphate backbone. DNA is actually two separate nucleotide strands. These strands come together and they are held together by hydrogen bonds. So the hydrogen bonds are the things that link the two strands together, and the linking occurs in the bases. Directionality of the subcomponents influences the structure and function of the polymer. Nucleic acids have a linear sequence of nucleotides that have ends defined by the 3' hydroxyl and the 5' phosphates. During DNA and RNA synthesis, nucleotides are added to the 3' end of the growing strand, resulting in the formation of a covalent bond between molecules. Remember that the nucleotide has three major components: the nitrogenous base, the phosphate, and the sugar. A nucleotide strand is a sugar-phosphate backbone. What you'll notice is it goes phosphate, sugar, and phosphate, sugar. When two nucleotides are connected, they are connected by a covalent bond. That is called a phosphodiester bond. What you'll notice in the nucleotide strand, it has a 5' end, which is denoted by the phosphate, and a 3' end, which is denoted by the sugar residue. Okay. So remember that. 5' phosphate, 3' sugar. DNA is structured as an anti-parallel double helix, with each strand running an opposite 5' to 3' orientation. Adenine nucleotides pair with thymine nucleotides via 2 hydrogen bonds, and cytosine nucleotides pair with guanine nucleotides by 3 hydrogen bonds. As I described in the last slide, the 5' end of the nucleotide strand is where the phosphate in. The 3' end of the nucleotide strand is the sugar residue, or the OH free available. What you'll notice on the right hand side of the slide is that when we have two nucleotide strands forming the DNA double helix, you'll notice that they run in opposite directions, such that one end goes 5' to 3' and the other one runs anti-parallel 3' to 5'. You'll also notice that our nitrogenous bases protrude into the middle of the residue. Those are like the rungs on a ladder. Adenine always pairs with thymine. And because of the nature of these residues, they always form 2 hydrogen bonds. Guanine pairs with cytosine, and these two residues form 3 hydrogen bonds. Hydrogen bonds are not very strong. However, when you have millions of these bonds being formed, DNA is a relatively strong molecule. The next macromolecule that we are going to examine is the structure of the amino acid. An amino acid has an amine group and a carboxyl group. It also has the side chain. There are 20 different amino acids, and these are differentiated by the different R groups. The side chains have a variety of biochemical properties. Some are nonpolar, some are polar, some are charged positively, and some are charged negatively. Now, just remember, you're not going to be tested on these 20 different amino acids. All you need to know is that each amino acid has a different side chain, and that provides the biochemical properties of that particular amino acid when they are used to build proteins. Proteins comprise linear chains of amino acids connected by the formation of covalent bonds at the carboxyl terminus of the growing polypeptide chain. In order to build polypeptide chains, we link individual monomers, which are the amino acids, together. They link in the process of a peptide bond. This is a covalent bond. As new monomers are added, they can only ever be added to the carboxyl terminal. So as the polypeptide chain grows, we always add it to the C terminal. Proteins are three-dimensional molecules, and their shape dictates the function of that particular protein. So we are going to examine the 4 levels of protein structure. The first level of protein structure is called the primary structure, and it's determined by the sequence or order of the constituent amino acids. So basically we start at the N-terminal and we add the individual amino acids one after the other after the other. And the order which we add them, remember there are 20 different amino acids, think of it as an alphabet. Each of the amino acids represents a different biochemical property, and we can add them in whatever order is necessary to produce that particular protein. These individual amino acids are held together by covalent bonds. So this is a relatively strong interaction between each amino acid. The second level of protein folding is called the secondary structure, and that arises through local folding of the amino acid chain into elements such as alpha helices and beta sheets. These two unique folds, the alpha helix or the beta plated sheets, these are caused by hydrogen bonds. And this occurs between the hydrogen of one amino acid and the negatively charged O of a corresponding amino acid. And so what we'll notice here is that there are certain regions within the protein that have these unique folds. Here are some examples of alpha helices and beta plated sheets, and you can see how they are localized folded structures. So these are just 9 different proteins, and you can see that the alpha helix is represented by that spring-like structure, where the beta plated sheet is represented by the arrows. The next fold is the tertiary structure. This comprises the overall 3-dimensional shape of the protein, and often it minimizes the free energy. This is where the protein becomes a truly 3-dimensional structure. There are several different types of bonds that occur during the tertiary structure. These include hydrophobic. Remember, hydrophobic is hiding from water. So these types of interactions are going to cluster towards the inside of the protein. Other ones include disulfide bridges. Sulfur is a unique atom present only in proteins. None of the other 3 macromolecules have sulfur. If you have 2 sulfurs near each other, they are going to form a disulfide bridge. Hydrophilic interactions, these particular amino acids are going to go to the surface of the protein. In ionic bonds, this is where you have positive charged R groups and negative charged R groups are going to interact with one another. The key thing I want you to understand about the tertiary structure is that these interactions occur via the R side groups of the protein. So the unique biological properties of the individual amino acid, this is where they interact with other side chains to form these 3-dimensional structures that the protein exists in. And the final structure of the protein, or the 3-dimensional structure, often occurs because it wants to minimize the free energy of the overall molecule. So in other words, it is the most energy efficient fold. The fourth and final structure is the quaternary structure, and this arises from interactions between multiple polypeptide units. Not all proteins have more than one polypeptide chain. However, some proteins have more than one, and this is where we get into the quaternary structure. 2 or more polypeptide chains are going to interact with one another, and these interactions are usually weak interactions, and they consist of hydrogen bonds, disulfide bonds, and these are usually between subunits that help stabilize the overall structure. So remember that these 4 elements of protein structure determine the function of the protein. You'll hear me use this term. Structure results in the function of a protein. In the diagram there, you can see how we have the folded protein where it's functional. When we unfold it, it loses its activity. And many of you have probably done this same experiment at home with an egg. We can see that the egg is water soluble. However, when you add heat to it, it now becomes insoluble. So you're actually changing the folding of the protein. You're actually interfering with the quaternary, tertiary, and secondary structure of that protein, and therefore you are changing the shape of it. This is just an overview of the primary versus secondary versus tertiary structure of the protein. You can see the compare and contrast at the different levels. So the third macromolecule that we're going to discuss is carbohydrates. These comprise of linear chains of sugar monomers connected by covalent bonds. Carbohydrate polymers may be linear or they may be branched. For example, the starch molecule is a linear chain, whereas glycogen, this is how our body actually stores carbohydrates. This is a branched molecule. This is in contrast to cellulose. This is the way that plants store carbohydrates. Their chains are much linear and longer. Animal cells versus plant cells, we store carbohydrates different.